and we wanted to use an organism that also can occur in these sort of areas, that's Caridina africana. So yeah, we just did a sort of standard protocol analysis to determine the LC50 for both atrazine and DDT, and then took it a step further to look at the LC50 for the combination of the two pesticides. So yeah, the two hypotheses for the individual chemicals was that atrazine would be toxic at sort of not environmentally relevant levels. We sort of based this on literature that we'd seen. And then DDT, obviously targeting sort of invertebrate organisms, we expected to be quite toxic to the test organism. But we then expected that there would be sort of a higher toxicity in combination of the two. For the results, though, we didn't really see that at all. For the LC50 values that we determined over the replicate exposures, we found that DDT and the combination LC50s came out very sort of similar, as illustrated by the sigmoid curves next to the results section. And so yeah, then we had to sort of accept the null hypothesis for our third hypothesis for the study. So yeah, that's basically it. Thanks.
Thanks, Greg. We'll come back to all the different questions again. Next up is Alicia Riley, looking at the physiological effects of contaminants in the Mediterranean mussels. Alicia.
Hi, can you hear me?
Yes, we can hear you.
Awesome. Hi, everyone. So in short, with the project, we just collected the mussel species Mytilus coloprovincialis along the coastline of South Africa to basically compare from site to site in terms of contamination. We were looking at biomarker analyses, so chemical and biochemical measures were taken to see basically if the animals showed any physiological responses based on their environmental stressors. We found no significant difference between the sites in terms of the individual biomarkers, but we did find that the Namaqua National Park obviously was our pristine site. Tsitsikamma and West Coast National showed more of a biomarker response. We then concluded in the end that Tsitsikamma is more or showed more of a biological response for the organisms compared to West Coast National, and we hypothesized more or less why that would be, or we theorized actually. Along the coastline of South Africa, there like a lot of inland pollution goes to the ocean. So although we did not link it to any specific contaminants, the findings were still seen, kind of. Yeah.
Great, thank you very much. Next up is Mbahlene Ngobeni on talking about hatching success and survival of Oreochromis.
Hello. Good day, ladies and gentlemen. My name is Mbahlene Ngobeni from the University of Johannesburg Zoology Department. So my study was based on the effects of the ARV tenofovir desoproxil on the hatching success and survival of the early life stages of Oreochromis mozambique. TDF, which is tenofovir desoproxil fumarate, is one of South Africa's ARV drugs used in the combination therapy. It was introduced after the phasing out of the ARV nevirapine, which had more adverse effects. So for my exposures, we had 4 groups: the control, solvent control, the lower as well as the higher concentration of TDF, and the exposure was carried out for a period of 30 days and replicated 3 times for statistical relevance. And then the results showed that there were actually no significant differences between the exposure groups that were repeated 3 times. In all the 3 repeats, we did not see any significant differences. Thank you.
Thank you very much. Next up is Kaylee Rainey talking about toxicity investigations of vanadium. Kaylee.
Hi everyone, thank you for having me. So this poster is basically looking at the effects of vanadium pentoxide in Oreochromis mozambicus. And this is part of my MSc, and it's only one part of it. I did LC50 studies before this, but this specific section looked at the biomarker responses that vanadium actually had on the physiological systems of the fish. And our basic findings were that vanadium, since it is a metal, does actually significantly increase metallothioneine contents within the organisms. There was also an increase in superoxide dismutase, which meant that vanadium actually creates ROSs within the organism that the organism is trying to decrease. There was actually a decrease in protein carbonyls, which is really, really interesting because it's supposed to link up with lipid peroxidation. Maybe its potential for vanadium is that it could decrease the effects of lipid peroxidation. And there was also an increase in catalase activity in very low doses of vanadium, but not in the higher doses of vanadium. So I actually suggest looking at chronic studies, because due to this being an acute study, it was only done for 96 hours. It could very well be that vanadium will increase the detrimental effects on the physiological systems of other fish species if left in, or if they are exposed to it for a longer amount of time. Thank you for listening and enjoying my poster.
Thank you, Kaylee. I'll just put you on the spot again for your second poster on the South African mussel species and their ABT temperature breaking points.
So this project forms part of my PhD, which is super, super exciting. It's actually on climate change. So this is the preliminary results just from Titicaca National Reserve. And I looked at the Arrhenius breakpoint temperature, which is basically increasing the temperature to a ramping point until the heart rate starts to decrease. And I'm comparing that between an invasive mussel species, which is Mytilus galloprovincialis, and a native South African species, which is Perna perna. And for my overall study, I wanted to see if Perna and Mytilus show differences, and if an increase in climate change, which one of the organisms would actually cope better. But in regards to this study, it was actually seen that Perna will not do very well in emerged conditions compared to their submerged conditions, and Mytilus will do better in their emerged conditions compared to submerged. And it makes sense because Perna is lower down on the shore, whereas Mytilus is a lot higher, and Perna is constantly being splashed and it's constantly coming into contact with water. So they won't be in emerged conditions for such a long time. And it's going to be very exciting to compare this study with this presentation from Titicaca with the West Coast National Reserve and Namaqua National Reserve. But otherwise, the study just shows that these organisms can have such a variable condition in both submerged and emerged conditions and between species. Thank you, Vaynand.
Thank you very much, Katie. Sounds like a really interesting study that you have there. So moving from the ecotoxicology to more on the biodiversity side, we have a poster from Anatoly Levinets on 3 Malumono species from South Africa. Anatoly, are you available?
Can you hear me?
Yeah, we can hear you.
Okay, good day, everybody. My name is Anatoly Levinets, and I am from Botany Department of Northwest University. In this presentation, we would like to report of 3 Malamonas species recorded in Vaal River. They are members of Cynophyta family of Chrysophyta or golden algae. Scan electronic microscopy of entire cells with intact scales and bristles allowed accurate identification of Malamonas carimbosa, Malamonas kenyana, and Malamonas portafera. You can see descriptions of these species in our poster, as well as some images and detailed morphology of them. The presence of Malamonas carimbosa in Vaal River represents the first record of this species in Africa. Current records indicate what Malamonas kenyana is restricted to tropical regions of the world, but its presence in temperature region of South Africa extends geographical distribution knowledge of this species. In contrast with earlier assumptions, what chrysophytes prefer waters low in nutrients with low conductivity volumes. Our results confirms what detected species are well adapted to warm eutrophic waters with high values of conductivity and neutral to alkaline pH values. Thank you very much.
Thank you, Anatoly. Then next up, moving to Zimbabwe, we have the diversity of aquatic macroinvertebrates from high altitude or a longer altitudinal gradient by Trevor Dube. Trevor, are you available? Yeah, Trevor, I don't think Trevor is here, so we'll move along to the next poster, which is Precious Iowa, looked at the molecular characterization of Lamproglena from Africa. Precious, are you available?
Hello, yes, I'm available. Can you hear me?
Yes, we can hear you. You can go ahead.
Okay, thank you, everyone. Thank you, SASAqS, for the opportunity. So with this research, we aimed at providing the first ever genetic information for the gill parasites of the cichlid and clarid fishes in Africa. We saw that even though these species have been reported a lot of times, there's been no information genetically for it. So we provided 18S and 28S RNA genes for it, and we've also been able to sequence or provide the CO1 mitochondrial barcoding gene as well. While we're able to compare with the other available sequences on GenBank from China, which is for Lamproglena chinensis and orientalis, our 2 species which we sequenced were the only ones in the globe for the CO1. So unfortunately, we are not able to compare it with other sequences. So we definitely are urging for more genetic information in this regard. And hopefully we'll be able also to do more phylogenetic analysis and compare the evolutionary relationships between this genus and maybe other carry out the higher phylogenetic analysis as well with it. So this is basically what the research was about. Thank you.
Thank you, Precious. I think the last 2 presenters are having some technical difficulties. So if you have questions for Divan van Rooyen and or Herman Morten Leroux, just drop them in the chat box and they will respond via the chat or on the YouTube comments. So if there's any questions for the presenters that just quickly highlighted their posters. I don't see any hands. Any questions? So I maybe have one question for Greg. You mentioned that the combination of DDT and atrazine didn't seem to have a higher response than the individual pesticides. Do you have any idea why that could be? Was there some sort of interaction or?
Thanks, Wynand. Yeah, I believe that there's no interaction, and that's why they're 2 completely different pesticides. One is a herbicide and one is an insects. So they will sort of operate in a different physiological way once sort of entering into an organism. So that's probably what we think, is that they sort of toxicokinetics and dynamics are totally different. We thought that the overall stress that the organism would be under the co-exposure, that's probably what would increase, if there were an increase, to change the LC50. But as I say, the atrazine seemed to have no toxic effect in combination with the DDT.
Yeah, thanks. Any other questions? Maybe I have a question for Precious as well. You said you sequenced 2 different species of Lamproglena, I think. How many different species of this parasite do we have in Africa or South Africa, more or less?
Yeah, thank you, Vainet, for that question. So we do have 13 of them in Africa and 6 in Southern Africa. We have, all of them are freshwater except the one Lamproglena lichii, which is the marine species.
Okay, cool.
Yeah.
Thank you. Other questions? I don't think there's been any questions asked on the YouTube playlist as well, or in the chat. I think the presenters did a really good job in their posters then, obviously. Maybe just a question for Alicia, maybe more of clarity. I think you said there was an increased biological response at the Tsitsikamma site, but compared to the West Coast and the Namaqua National Parks. But I didn't hear if you gave any reasons for that, or.
Hi, yes. So Tsitsikamma is located close to the East Coast, where 90% of the estuaries actually drain into the ocean. So it is presumed that a lot of the contamination that happens inland are draining out into the ocean. Also, there is the harbour. Similarly, the West Coast National is located close to Cape Town, as well as the Saldanha Bay harbour. So it is presumed, due to the population surrounding that area, that the pollution is going to be a little bit higher than in Namaqua National Park. Namaqua is located higher up, where there is limited population in the area.
Okay. Any final questions? Okay, then I think we'll conclude this session there then, if there's no other questions. So please, everyone, go and watch these posters and read these posters if you haven't had a chance already. If you haven't had a chance to watch all the presentations that's come today, please go and do so. Watch them, give some comments, feedback, everything is welcome. Please also like the presentations that you feel is deserving for the best presentation. That's that award as well. So then a few things for tomorrow, just to highlight, and there's been one or two gremlins that crept into the program. So just to remind everyone that tomorrow's program will start at 10 o'clock with the keynote presentation. We've specifically arranged it so that you have time tomorrow morning with your cup of coffee when you get to the office or wherever you attending the conference, in front of on your couch, wherever, to have a look at all the presentations that's lined up for tomorrow. There's quite a lot of interesting things coming up, specifically on fish biology and ecology. So please go and support all of the students and presenters and get all your questions ready. It really helps with the discussions if there's some interesting perspectives that you would like to bring forward. So the room will open again at about 9:15, 9:30 tomorrow morning for you to log in, get ready, for the session then at 10 o'clock tomorrow morning. Also remember, if you haven't registered yet for the medal ceremony tomorrow at 4 o'clock, please do so in the link that I've shared in the chat as well as by email. It's on Zoom, please remember that, so you have to follow the same link that you register again for the session tomorrow at 4 o'clock. If no one has any further questions or discussions, then I'd like to end off today. Thank you very much for all the presenters, the chairpersons. Enjoy the rest of your afternoon, and then we'll see you tomorrow morning for day 2 of SASAqS 2021.